So this morning, I have the pleasure of introducing our plenary speaker, Professor Fiona Hunter from Brock University in Canada. Professor Hunter has a PhD from Queen's University in Kingston, Canada, and has a career focus on biting flies, principally black flies, but more recently on mosquitoes, virus transmission, and so forth. And she tells me that she likes working on biting black flies better than mosquitoes. But more recently, since 2001, she's been working on West Nile virus mosquito surveillance and virus testing in Ontario. And some of her current research includes studying co-infections of viruses such as combinations of West Nile virus, Zika virus, and Mayoro virus. Yes. And then also, actually more interestingly to me, insect-specific flaviviruses that have a potential to block transmission of pathogenic viruses. So, just going to advance it there. Get started for you.
Good morning. Today I'm going to talk about some of the work that we do on mosquitoes, even though I don't particularly like them, as we have just heard. And obviously I have to thank my research team who have helped with this. So shown in the photograph are all of my students and postdoc, Dr. Larissa Borelli. She's the bottom left. And also I have some other people who work with me. I have a company off campus called Entomogen, and my manager there, Alessio Gasparato, is one of the best mosquito identifiers I've ever had. And he's been with me for over 20 years as well. And then former PhD student Brian Giordano, he had his fingers in this as well, and he's now down in Florida. So I'll give you an overview of what I'm going to talk about, titling it, looking at co-circulating viruses. But really, I want to tell you what one does with the same old type of work for 20 plus years, which is what the mosquito surveillance and arbovirus testing in Ontario has been. So 20 years, it was research in the beginning because we didn't know who was transmitting or what species were transmitting. But now I'm really searching for novelty because it gets a little old doing mosquito surveillance and viral testing. So I'll tell you of some of our exciting finds. One of the things, of course, is range expansions, because we've got an amazing database of what species are there and when they come into the country, come into Canada, and just basically what surveillance for arboviruses is. And if you don't know where Ontario is, the map shows you in green. That's the province that I work in. And then I'm going to switch gears and get to the year 2016, 2017, because by then we'd really figured out everything that was going on with West Nile virus, in my opinion. And so we had to do some other sort of work. And that's when we switched to doing a lot of vector competence work, and specifically for Zika in our containment level 3 lab at Brock University. And then, after we've got all that information, the next stage is, let's try and keep the excitement up and do something else. So started looking at the possibility of co-infections in mosquitoes. I don't really care what happens in humans if they get co-infected with 2 different viruses. I know that's heartless, but I want to look at it from the mosquito's perspective. And so I have a couple of examples of co-infections I'm going to talk to you about. So there we are, 1999, West Nile virus hit in the borough of Queens, down in New York City, where crows and other birds started dropping dead out of the sky. And shortly thereafter, I got a phone call from the Public Health Agency of Canada saying, hey Fiona, can you please help do mosquito surveillance? for West Nile. And I'd been a black fly person. I loved my black flies. I loved going collecting with Monty Wood and Doug Curry and all those people. And I said, okay, I'll do it for a while. And I've been stuck in mosquitoes ever since. Anyway, what it means is I get access to samples from all across Ontario of mosquitoes that have been trapped, primarily using CDC light traps. And Ontario is a very big province. We have 35 health units, as shown here, all the different health units. The luxury that I have is that I don't have to go to all of these places to get my samples. The public health people set the traps, send them to us, we identify and do the viral testing. So probably, we're well about 6 million individual mosquitoes that we've identified and viral tested over 20 years. It's an enormous data set. And I just want to show you one little bit on this map. Down here, this is going to be Windsor, Ontario. I'm going to talk about Windsor in a few slides. So we've got data from all across Ontario for well over 20 years. And so in that time, let's just not do the same old identifying and viral testing. We came up with a photographic key to the adult female mosquitoes of Canada that's been very successful in that Canadian Journal of Arthropod Identification. We've taken all of the information, all the really good identifications that we had, and we were involved with Polly Bear back in the day. This is one of the first big papers of barcoding mosquitoes. That was back in 2006, yes. So I contributed to science. And most recently, okay, now this is really exciting for me. I have a grad student who wants to work on mosquitoes, but he really likes the oddballs, and he's doing a lot of field work for Culicidae perturbans, and we're actually trying to get a Culicidae perturbans colony set up at the university. And he's been able to rear them. He's having great success. And he found the pupae, which are absolutely spectacular. You'll probably know that Culicidae perturbans has a piercing siphon for the larvae that sticks into aquatic roots and shoots to get its oxygen. Well, the trumpets of the pupae also do that. And on the right-hand side, you can see that those trumpets are perforated, or they've got this saw-like edge to them. So when they're disrupted and moved, they actually can move these trumpets independently of one another and try and stab their way back into the aquatic roots. Spectacular. This is one of the reasons I'm still doing mosquito work. All right, so then, all this data, what have we also found? We've got new species to add to the species checklist for Ontario. And everything in red here is a new record from when we started back in the early 2000s. I'm not going to go over all of them, but I do want to highlight some of them that we actually saw in real time creeping up from the United States into Canada. So we're going to look at Japonicus, Culex erraticus, and then Aedes aegypti and Albopictus, which never in a million years did I ever think that they'd be coming to Canada, but they did. All right, so we'll start with Japonicus, absolutely gorgeous creature. Way back in 2001, when we were just starting off and we weren't yet identifying 300 some odd mosquitoes a year, we only had 42,000 that we identified that year. We found a single specimen of this thing that we didn't know what it was. Turned out it was Japonicus. 2002, we found 9 specimens. And they had moved from our health unit, which happens to be the Niagara Health Unit, to a neighboring health unit. The next year, 22 of them showed up in these traps, now from a variety of health units. By 2004, they were in 22 of the 35 health units. They just exploded, and I could keep going, but I'll tell you, they are now the most commonly collected container breeding mosquito in Ontario. And we saw them in real time creeping their way up and expanding, which is in itself, pretty exciting. The next one, about 2008, I think it was, we found 5 specimens of odd Culex for us. Turned out they were Culex erraticus, which is also a potential vector for a number of diseases. And we kept looking and looking and looking through our hundreds of thousands of mosquitoes each year. Found a few, so you would read this from the bottom up. Where am I? So just a few specimens for the first few years, then they start to increase. And the number of sites that they were collected at, just a few, and now, hundreds of sites. They're never very common, but they're definitely established in Ontario. And so now they're widely distributed across southern Ontario. And really, we saw them in real time, marching forward or northward. But really, one of the most astounding things for me was in 2016, where we found both Aedes albopictus and Aedes aegypti in a lot next to a train depot, a commercial train yard. And we found larvae and adults of Aedes albopictus and Aedes aegypti. And, I thought, well, obviously they're coming in on the trains. At least that was my assumption. Never in a million years thought they would come to Canada. Well, here are the numbers. So we're still, Aedes aegypti is still just an occasional incursion, probably coming up on that rail line, because it's not actually changing its position in Windsor. It's definitely not overwintering. And I can say, we have been down into sewers in Windsor looking for evidence of overwintering for aegypti. We've been in the tunnel that connects Windsor to Detroit looking for Aedes aegypti potentially overwintering. So we're pretty sure this is just an occasional incursion that happens. And we haven't actually seen them since 2019. But we have to keep looking for them. But Aedes albopictus has, in fact, established shop, or is now actually living in Windsor, Ontario. We now have 24 sites that we survey every year. We haven't found them yet this year, but it's usually a little bit later in the season. And I have no doubt whatsoever that they are established. So, where did they come from? If you look at this little yellow dot here, that's where Windsor, Ontario is. And as I say, I'm fairly confident that they are established. We find them throughout the end of the season, we find them early in the season from year to year, and we find adults and larvae at different places. All still within the Windsor area. So they're not actually spreading like the other ones did, like wildfire out into other parts of the province. But where did they come from? Well, we know that there are established populations, at least when we're looking back at 2016. There are already established populations of Aedes albopictus in bordering states. Interestingly, in Michigan, the year after we found them, they found them. This is from news sources, by the way, but only ever a couple of specimens, and that was 2017, 18, and again in 2020. So what I'd really like to have is specimens from all of these states to see if we can do any genetic analyses to see. So that's what we did. Brian Giordano, my former PhD student. This is not published because it's a work in progress. I've color-coded here using the purple for the Windsor specimens and a blue color for Ohio specimens. And we definitely see clustering of Ohio and Windsor specimens. Obviously, we need a lot more information to see if we can really trace where they came from. And again, I'm grateful that they're not moving really quickly because we still have time. All right, so I'm going to switch back to the ho-hum mosquito surveillance that we've done all these years for West Nile virus. We use a variety of traps. CDC light traps are good for a variety of species. Then we have the BG Sentinel traps that are good for anthropophilic Aedes species. We use these really heavily in the Windsor area. And then we even do the dirty gravid traps to try and get blood-fed specimens. And all of this goes into our 6 million specimens that we've been looking at. And we've tested for West Nile virus. We've tested very regularly. We test 13 different species. And I can say without a shadow of a doubt that in our area, Culex pipiens is the driving force. If you don't get infection in Culex pipiens, which is of course doing the bird transmission, then you're not going to end up with human cases later in the season. I said I didn't care what happened to humans, but this is public health type work. So all this work culminated in a paper we had in 2017 where we can actually predict now how many human cases there are going to be. And this quadratic equation is highly supported. R-squared values of 0.97. Along the x-axis, the number of positive Culex species pools that we get in a given year. And because we have the same emphasis on surveillance year after year after year, we can do this. It's not as if it's once in a while we do surveillance. We just keep hammering away at it year after year. And on the y-axis, that's the number of confirmed human cases that we find. Beautiful equation, and we can go through the season and tally up the number of Culex positive pools that we're getting, and that can, then we can tell the health units, watch out, this is going to be a bad year, you're going to get a lot of human cases, now's the time to do your public messaging, tell people not to get bitten by mosquitoes. And if it's very few specimens, then we know that we are going to have a good year, not a lot of human cases. But what else was going on in 2016 and 2017? Because honestly, I thought I'd done as much as I possibly could with the arbovirus surveillance. You may remember that in 2015, there were about 7,000 cases of illness in Brazil, rash, conjunctivitis, and so forth. They didn't test for anything at the time. They certainly weren't testing for Zika, but by February 1st of 2016, the World Health Organization had actually decided that there was a problem with Zika and the clusters of microcephaly that were occurring in Brazil. And so the next month, I hustled down to Maceió, Brazil, to a summit on Aedes aegypti, and it was called the Summit Aedes aegypti Crisis in the Americas. And when I got there, I discovered everybody was already saying Aedes aegypti is the big vector for Zika virus, we've got to eradicate it, etc. And when I asked the researchers there, nobody had done any testing of Aedes aegypti. They had no evidence whatsoever from surveillance that it was testing positive, which, after my 15 years, 16 years of doing West Nile surveillance, I thought this is preposterous because we know that there are other species that can transmit things. So I came back and I started looking at the Flaviviridae. So I didn't realize this, but the virus people, they italicize the family name for viruses, and unlike us. Anyway, so I looked at the Flaviviridae, that was just an aside, and this is a paper from Moreau in 2015 looking at the evolution of the flaviviruses. You know, I can read one of these because I had a good background in systematics and phylogenetics, et cetera, and actually say that the ones at the top are Culex associated and the ones at the bottom are Aedes associated. And where does Zika fit in here? This little dot here, which doesn't really seem to line up in my opinion. So what did I do? I went and took that paper, which had 53 different species of flaviviruses, and I extracted ones that I'm familiar with and to make a nicer, smaller tree. So I looked at St. Louis encephalitis virus, West Nile, Zika, dengue, yellow fever. And this is how they are actually separated in that paper, and this is how the International Committee on the Taxonomy of Viruses thinks they should be separated. They clearly state that Zika is Aedes associated. But is it really? So, this is my much simplified version of the phylogeny. And there's a node here that's supported 97% that clearly clusters Zika with St. Louis and West Nile. Even though everybody in the virus world and many, many people in the public health mosquito world just accept that it's Aedes associated. And when I looked at the disease that happens, the little brains show that that's neurotropic effects. That's West Nile, St. Louis, and Zika virus. The blood drops, that says that it's hemorrhagic type disease. And then looking at the types of reservoir hosts. The little birds, obviously avian reservoirs, and the little primates are primate reservoirs. So, you know, I propose that really we should be looking at other mosquito species to see if there are other species that can transmit virus. And incidentally, I got heckled at an ESA conference for suggesting this. Anyway, there are people who have really good evidence now from Brazil that Culex quinquefasciatus is a good vector of Zika virus, and there are also examples from China. But, I mean, we're back in 2017, right? So came back, we started doing some vector competence studies, and how we do that, we infect the mosquitoes, keep them in a containment level 3 lab, which we happily have at Brock University, and 10 to 14 days post-infection, we rip off their legs and wings, keep those separately to test them for a disseminated infection. We stick the still living mosquito, minus its legs and wings, with its proboscis in a microcapillary tube, and we collect its saliva, because if there's immersion oil in that microcapillary tube, it'll actually spit into it, and you can watch them spit under the microscope, which is pretty exciting. And then afterwards, we take all these body parts and extract the RNA and run it with qRT-PCR to see if we have any virus. So as I say, this is way back in 2017. Former master's student Shay Shivafard did this for me. We found, using wild-caught Ontario mosquitoes, the only one that was able to get infected and transmit. So IR is the infection rate, and TE is the transmission efficiency. The only one that came up positive was Culex pipiens. And then we had colony mosquitoes, so we tried those as well. And it turned out Albopictus was way better in our hands than Aedes aegypti was. And if you follow the literature now, yeah, there are a lot of other species that can transmit. And even the early literature had another huge whack of species that were known to have tested positive. So really the paradigm that it's only Aedes aegypti should be tossed, in my opinion. All right, So look at this, Culex pipiens, also a major West Nile vector. And this is when I thought, okay, now we can start doing some real science again and look at co-infections. And that's what we did. So we've been looking at the possibility of two flaviviruses inside a mosquito, what's going to happen. Or we take a flavivirus and an alphavirus, put it in a mosquito, and what's going to happen. And this is where we use West Nile and Zika as flavies, and then Zika and Myarovirus as our Myarovirus as our alphavirus. And that's because one of my other PhD students is from Brazil, and Myarovirus is very common in Brazil. So we're trying to do things that have real-world relevance. And usually we start with cell culture studies first to get a handle on this. So in our two flavies, when we test West Nile and Zika in cell culture, okay, I'm going to walk you through how to read this. It's a bit ugly. We're just going to look at these bars. The solid red is a viro cell culture, that's a mammalian cell culture, and the solid blue is also viro cell, but red is West Nile and blue is Zika virus. And across the x-axis, you're looking at days post-infection in cell culture. And all you really have to see is that the red bars, which are West Nile, are taller than the blue bars, which is Zika virus. And the conclusion is that in cell culture, West Nile virus wins in mammalian cells, that it replicates more. But I'm not interested in what's happening in mammals or humans. So let's look at these other set of bars. These are in E636 cells. It's a mosquito cell line. It's actually from Aedes albopictus, but this is our mosquito cell line that we work with. And the same thing, red is West Nile. This time they're hatched, so they look darker, and blue is Zika. And there is absolutely no statistically significant difference as we go from day 1 through 2, 3, 4, 5 in the amount of virus that's replicating. And that tells us that there is no winner in mosquito cells. Now we're going to have the possibility of something exciting. We're going to see what happens when a mosquito is infected with both. What's it going to spit out, or is it going to spit out both of them? So that early work that we did with Zika, as I say, was 2017. My current team, shown on the top left, smiling because everybody was so happy. Larissa Borelli, my postdoc, Amanda Williams, a master's student, and Yvonne Drahoun, my PhD student. They said, we'll do it, Fiona, and have spent months working on co-infections in mosquitoes. This all has to be done in a containment level 3 lab. You have to be dissecting these mosquitoes. They're potentially infectious. You have to do it in a hood, with your arms in this box. And it's very, very time consuming, picky, and we have to account for each and every mosquito that is in the CL3. They're all so happy. What a great group. So we looked at West Nile and Zika in Culex pipiens because we know from previous work, West Nile is in pipiens. It's one of the major vectors. And we also know that in our hands, we could get Zika to be replicating in Culex pipiens. So on the left-hand side, that shows you the number of individuals, and it takes a long time to do all this for each individual. So 24 that were given West Nile only, 10 that were given Zika only, and 12 that were given both of them in a co-infection. And when we look at the results, well, only 4% were positive for West Nile, which is ludicrous because they're a major vector. 20% were positive for Zika, which, well, that sort of supports what we said back in the day. But 85% of them were actually able to spit it. That's the transmission rate at the far right in the middle there. Only 7 of them actually spat. As I say, you can watch them spit. So that number isn't, the full 10 specimens. And what happened in co-infections? Absolutely nothing. Nobody got anything. And all of these are totally unexpected results. So I said, not to worry, we'll continue, we'll come back and revisit that. They're actually in the lab this week revisiting this for me. And we'll look at the flavivirus and alphavirus co-infection, Zika and Myaro. And this is work Amanda Williams has done in cell culture. Myaro alone has a really nice curve. So what this is, the log starting quantity. This shows you how much more virus is actually being produced. This is days post-infection. And Myaro alone and Myaro in a co-infection have the same amount. So Myaro is not affected by a co-infection with Zika in our C636 cells. I'd just like to show you the value here is 8 for this log starting quantity. So they do really well. If we look at Zika and Myaro in the, Zika and Myaro in the co-infection, but now we're just going to look at what was happening with Zika, we can see that Zika on its own had a log starting quantity increase. It was up at 6, whereas Zika in a co-infection is way down at 4. So Zika is negatively affected by a co-infection with Myaro in C636 cells. And the number down here, it's just 4 as opposed to 8. So if you put all this together in a co-infection situation, Myaro's winning. So we sort of expected that would happen in our infections. And when I was writing up the abstract for this talk, we knew that we had done a quick and dirty study with 9 Zika Myaro co-infected individuals. We expected Myaro to outcompete Zika, and we ended up with infection rates of nobody had just Zika, nobody had just Myaro, but 2 of them had both viruses in them. So I thought, oh, this is interesting. Let's continue in this vein. So my really, really happy team went in there. We increased the sample sizes. This time we're looking at Zika and Myaro. Zika only 26 specimens, Myaro only 22, Zika and Myaro co-infections, 20 individuals. And the team became less happy because this is what we got. Nothing in Aedes aegypti, I should say, we're now looking at Aedes aegypti, because that really would be a real-world situation down in South America. Nobody was positive for Zika virus. Only 1 was positive for Myaro, but it didn't spit it out. And then of all the co-infections, 1 had Zika only and 1 had Myaro only. Nobody's getting infected. Months and months of prep and work, and nobody's getting infected. And so now we are thinking we've got a problem, and that is that we recently hired a new virologist who uses my insectary, well, it's now our insectary, to raise his mosquitoes in, and he works on insect-specific viruses. And these insect-specific viruses can protect mosquitoes from arboviral infections, as he showed in a lovely 2020 paper. And so we're now going to have to go back, science marches forward. We now have to go back and see maybe all our stocks are contaminated with insect-specific virus that is protecting them from infections, and our co-infection studies we can just toss out. Or it's going to really inform that we can use this as a control strategy. So things are beginning to get a little bit more exciting again in the mosquito world. And that's all. Thank you very much.
Question?
Fiona, how much coordination is there between health authorities on both sides of the border?
We have to make our own, we have to make our own connections on the two sides of the border. So we have somebody in Ohio who's more than happy to share specimens with us, but we haven't managed to get that in some of the other states.
Yes, go back.
When you do those co-infections with the two viruses, do you see any chimeras with viruses pick up?
We haven't actually looked at that. We should.
Have you considered tourist agents or either visitor of people coming back from holidays by car that these might actually contain these because that's the situation.
We've considered that it was people bringing them across the border, but the fact is that we have now these 24 sites that we survey, and year after year they turn up in those sites. So I think it would be unlikely that the tourists are bringing them back to the same sites, and they're all sort of industrial sites.
Thank you for this talk. It is really a difficult field because, you know, there are a lot of limitations with the cell cultures, the story of the cell culture when you're exposed to different viruses. The colonies is also very difficult as you explained it there. So do you have any collaboration with other teams where you can go to the field and try to use field colonies which are recent, maybe not so long story as the ones you have, really try this kind of co-infection?
We collaborate very closely with the National Microbiology Lab in Winnipeg because our two labs are the only ones that have the CL3 with insectaries. And so we do collaborate with them, but it's still a work in progress.
I'm thinking more in endemic countries.
Oh, in endemic countries.
Because they have the material vectors which are really...
Right. I actually work quite often down in Dominican Republic. I have a little lab set up down there. We don't have a CL3, but I can at least get material from them and bring eggs back. Yeah. Thanks.
Sorry, let me see. Thank you very much. The next